So there we are. We are recording now. Some other things before we start. Once the webinar begins and I hand everything over to Darko, you will be muted and the video will be disabled. Feel free to put any questions in the chat. They won't be answered until the Q&A section of the presentation, but we will absolutely get around to them. During the Q&A, you should be able to unmute and turn your video on if you want to ask any questions with your face and with your voice instead of just typing it out. And after this webinar, we will be sending out a feedback form to, learn more about what worked and what didn't, and all of that. So please fill it out. Also, this is my last webinar with the HETF. So we don't have another one scheduled, but expect that this webinar series will continue sometime in January or February. So with that, I would like to introduce Darko D. Cotoras, who is a research associate with the California Academy of Sciences. His main research interest is on the historical processes that create biodiversity in conditions of isolation. He focuses on invertebrates from islands in the Pacific Ocean. He did a postdoc between the California Academy of Sciences and the University of California, Santa Cruz on improving ancient DNA methods for museum specimens, techniques which were applied on island biogeography of spiders. Sponsored by a Fulbright Fellowship, he earned a PhD on the Integrative Biology Department at the University of California, Berkeley. Here in Hawaii, he studied the temporal dynamic of the Tetragnatha spider's adaptive radiation. And with that, I will turn it over to Darko.
Well, thank you very much for that introduction, and especially thank you very much for the invitation. For me, it's very special to have the chance to share the results of this work with the community from the Hawaii Experimental Tropical Forest and everyone else invited to participate. It's very special because many times you do research and the people with whom you interacted during the development of that research might never know what you did. So it is great to be able to be here in person or as close as you can in these COVID times to many of the people who helped me directly to accomplish this fieldwork. So with that big feeling of gratitude, I would like to start telling you about what we're going to discuss today. So today the talk has two big segments. Like the first half would be a description of the geologic history of a volcanic island, focusing, of course, in the Hawaiian archipelago. Will be more or less a rather long introduction to place in context what will be on the second half, where I will focus on a group of endemic spiders and I will test hypotheses that show how this geologic landscape, this dynamic geologic landscape of the Hawaiian Islands serves as a pump for species formation. So the first part might be for some of you that live in Hawaii or even work directly with the landscape, might be perhaps basic. So I apologize for that, but I think it's still important to put everyone on the same page so we know what are the processes, these forces that are shaping the biodiversity of the islands. So to place this on a biological context first, I will talk about adaptive radiations. Adaptive radiations are perhaps one of the most interesting phenomena in evolution. They correspond to the rapid diversification of a big number of species in a very short period of time. And associated with this diversification, there are some ecological axes which allow those species to partition the niche of where they live. So many close related species are living in the same place, but doing different things. We find adaptive radiations across all the planet in all branches of the tree of life, vertebrates, invertebrates, plants. We find adaptive radiations on long temporal scales like millions of years as the marsupials in Australia, or hundreds of thousands of years more quickly like cichlids in East Africa. Adaptive radiations tend to occur in isolated environments such as islands, mountaintops, lakes, and that is why volcanic islands such as the Galapagos and Hawaii are perhaps the capitals of adaptive radiations, where we find this phenomenon repeated over and over again across many taxa. And I have to say that, of course, Hawaii is one of the top places where this has been studied the most. Now, within the understanding of adaptive radiation, there is a gap in knowledge related with what happened at the very early stages. We know that they diversify quickly, but how does it work biologically? So to address that, one option would be to have a time machine and travel back in time and sit there and see how species evolve. We cannot do that, so we need to use different proxies. And to get an idea of how those proxies work, I invite you for a second to don't think about islands or spiders, but think about a metaphor. Imagine that you are a minister of education in a country, and you implement a reform in education, and you want to see what are the effects on the students. So one very traditional way to assess that would be to, after implementing your reform, give the students a test. Then you see how they perform in first grade, then in second, then in fourth, and then once they graduate, you go and talk to them in person. So once you talk to them in person, you can gather more information than just their poor mathematical skills. Your reform didn't work really. You can gather information about what they know about history, about literature, about philosophy, etc. So this action that I just described is very similar to what paleontologists do. The exams are really the fossils, and the extant species are like the newly graduate students. So as a paleontologist, you can see how a species changed through time, how a student changed through time, and then you can investigate more directly what happened with the current species. This work when you have long time scales and when you have good fossilization conditions, which usually is not the case for volcanic islands. So then there is a second approach, which is called a synchronic approach. And this one could be applied when you are asking a question about a process that is very stereotypical, that happens over and over again, where you don't need to follow the same individual through time, where you don't need to follow the same species through time. In our metaphor, for instance, would be our cognitive development. Everyone, when as a kid, is very curious. Then as a teenager, you don't care about school maybe. Then you find what you're interested in and you become passionate about it, and then you graduate. That happened to most of us. So you don't need to follow the same person. You could go to the school and interview with the cohort that is in first year, with a different cohort in second year, all of them going through the same process. This is a chronosequence approach, and chronosequence approaches have been widely applied to study volcanic islands. So I think for this crowd, I don't need much introduction to the Hawaiian archipelago, but in case someone is not living or working in Hawaii, I will briefly talk about it. This set of islands are in the tropical North Pacific, in halfway through between Asia and North America. They start down here with the Big Island, then they extend to up to Kauai with the main Hawaiian Islands. Then we have about 3,000 kilometers of what is known as Papahānaumokuākea, or the northwestern Hawaiian Islands, where you find shallow reefs, rocks, and little atolls. And beyond beyond Kure in the northwestern Hawaiian Islands, we have a long underwater mountain chain that, after these change in directions, end up subsiding under Russia in Kamchatka Peninsula. So Hawaii is really way longer than what you see on the main islands. Here, a zoom in, including the main Hawaiian Islands and the northwestern Hawaiian Islands. These islands are what is remain after. These islands are the remain of bigger islands that at some point were further south, once the Pacific plate, before it moved so much in that direction. So this volcanic archipelago is produced by a stationary hotspot that currently is in the south of the Big Island. Lo'ihi and Pu'o crater are the most active areas, and this is constantly expulsing lava while the Pacific plate is moving in a northwest direction. So like a conveyor belt, islands are produced and then carried with the plate. If we focus on the main islands, here we have Big Island, Maui Nui, Oahu, and Kauai. And these will be the main characters of this first half of the talk. So now let's do a very quick volcanic island geology 101. So these islands start as underwater volcanoes. Around Hawaii, the waters have near 6,000 meters of depth. So once you see a tiny rock sticking out of the ocean, that is the tip of a volcano 6,000 meters already. I will talk about the Big Island and highlight three different geographic features that will allow us to understand more of how this geologic life occurs on these islands. The first one is, of course, the areas that are actively growing right now with volcanic activity. So when the island is producing this lava, new land is generated, but this land is very poor in nutrients, low phosphorus, low nitrogen, low calcium. All the nutrients are encapsulated in the rock, and after weathering, they will be released and produce more nutrient soils. In the case of the Hawaiian volcanoes, as well as other volcanic islands such as Marquesa Society, Easter Island, Juan Fernandez, etc., this lava is different to the lava that you find on volcanoes produced by subduction, such as the Andes or the Cascades in North America. These lavas in the volcanic islands are lavas rich in iron, coming from very depth magmatic chambers. In the case, magmatic chambers, right? And that is in opposition to the lava that you find in volcanoes produced by subduction, where the lava is really melted oceanic floor. So rich in silicium, more water content, so then the lava is more explosive. In the case of the Hawaiian volcanoes, the lava is not as much explosive and tend to have a higher viscosity. It is so high that sometimes it could solidify, showing all these faults of the liquid as it's moved on the landscape. This type of lava is called the pahoehoe, in opposition to the a'a, which is a lava that tends to be produced in more steep areas, and this lava is more like what you would call a volcanic rock. One of the features that is very interesting and interacts with the biology is the kipuka. Kipuka are the names of these islands of forest that are produced by lava flows. In this case, it's a satellite image of the saddle area between in the center of the Big Island, and we can see a historic lava flow from 1845 or 35. And these little pockets are islands of vegetation that are formed instantly, geologically speaking, of course, once the lava goes through this landscape. This idea, this setting called the attention of many biologists in the past, especially Hamd Carson, who referred to this kipuka as one of the sources of diversity on the Big Island. Initially, it was thought that probably this isolation might produce local species, but there's not much evidence to that. Yet this dynamic of separation and later admixture once the forest regrows over this volcanic area might create an increase on genetic diversity. And that's why Carson calls the Big Island a cradle of evolution. Another important feature of the Big Island, especially associated with its massive size, is that it can reach elevations of 4,300 meters. These high elevations are high enough that seasonally there could be snow. So you can imagine that a place where there's coral reef at sea level and snow at the top in a very small area will compress a huge amount of biomes or ecosystems. And that is another driver of the high diversity in the Hawaiian Islands. Also, a volcanic island grows very quickly. So the oldest volcano on the Big Island has about 0.5 million years. And that time is enough to reach these very high elevations. Mauna Kea is even younger. But after a very quickly buildup, associated to a very quick buildup, there is very low complexity. So on the Big Island, we can see, especially in Mauna Loa, very gentle slopes, very simple topography. And also when the islands are young is when they reach their biggest area. So very quickly they grow, and then the next 5 million years will be a slow story of subsidence and erosion. So here, a geologic map where you can identify very clearly the 5 main volcanoes of the Big Island: Mauna Kea, Mauna Loa, Kohalas, Walalai, and Kilauea. Now, if we move up north, we see Maui, where we have two volcanoes, West Maui and Haleakala. The two features I want to focus are first the Haleakala caldera and the Ko'olau gap. Haleakala caldera is about 3,800 meters, so very high too. It is called a caldera, but it's really a summit that has collapsed because it's above the inversion layer. It's very dry, which you would, in principle, wouldn't associate with with the rainforest down below, and is dominated by different cinder cones. There are also these gaps like the Ko'olau gaps or the Kipahulu Valley, which are examples of deep erosion, a feature that you only see on the young island of the Big Island in the very north of the Ko'olau, of the Kohalas, with Waipio and Waimanu Valley. But you don't see that in the younger volcanoes. So this is one example of how age adds topographic complexity. And associated with topographic complexity, we will have species production. As part of the Maui Nui complex, we have the island of Molokai, and has this very strange shape. Well, it's not strange, but it's curious. It's like a rectangle. And if you look at closely in the south side, you will see all these ravines, which they don't seem to have a mirror image on the other side. Instead, there's this clear cut associated with very steep cliffs that it's produced by a massive landslide that eventually happened in taking half of the Molokai down to the ocean and producing a mega tsunami, for which one there are evidence in other islands. So this rapid collapse or this landslide is associated with the fast growing pace of these volcanic islands. Also, when the islands get older, as I mentioned, there is a buildup of soil, so nutrients increase. And with associated, well, independent to nutrient increase, because these islands are so massive, they deflect the bottom of the ocean and they start subsiding. So they are literally sinking as they age. Lanai, also part of this Maui Nui complex, it's on the rain shadow of the volcano on Maui, so it's drier. And Kaho'olawe is smaller and even more dry. Another interesting element of Maui Nui is that if we go back in time 1.2 million years ago, it would look much like what we see today on the Big Island. So a bigger island with several volcanoes all connected by land. But as time passes, as you can see here, the island starts sinking and space in between them gets filled with water. Now, this dynamic of fragmentation gets interrupted at a smaller time scale, at the time scale of the ice ages. And during ice ages, with changes on sea level, some of these islands get reconnected temporarily for time scales of tens of thousands of years. So the same way as in the Kipuka, we see dynamics of admixture and, separation and admixture in time scales of hundreds of years. Here we see separation in millions of years, and intermittent admixture and separation due to eustatic changes, so sea level changes. All of these factors, as you can imagine, adding to increase biological diversity. Moving up north, we have Oahu with two main mountain chains associated with two volcanoes, the Waianae and the Ko'olau range. Here I'm going to talk first about the Ko'olau Mountains as a prime example of water erosion and landslide, producing these really shape-laid mountain chains that are characteristic of volcanic islands. You could see the similar pattern, similar structures in other islands like Tahiti or in the Juan Fernandez, in Robinson Crusoe, as well as the Marquesas. These are tropical islands, of course, and there is another element building up with time, which is coral reef. Coral reef builds on the island plateau, equivalent to a continental shelf, but for an island. And in many cases, after the island completely subsides, the ring that forms the atoll is the coral that is on the edge of the island, and give us an idea of what was the size of this older island. This is very clear when you look at some of the northwestern Hawaiian islands. In the case of Hawaii, there is less coral development than other places in the South Pacific for several factors. One of them, Hawaii is located around 20 degrees north, so it's not right on the tropics, so maybe temperatures are a little bit lower, but also many of these islands have very steep sides, so that makes that the photic zone, the area where the corals can grow, is very narrow. In the case of Hanauma Bay, there is a development of fringe reef because this is a crater that is open to the water. Now, Hanauma Bay, Coco Head, Diamond Head, they're all part of the same type of geologic phenomenon, which I haven't mentioned yet, which is volcanic rejuvenation. Some of these volcanic islands would have later volcanic activity associated with magma chambers that did not become active until later on life. So this is, you can call it some sort of like a second air that these islands get on their middle age. And then moving on to the oldest of the main Hawaiian islands, Kauai. Some traits that I want to highlight here. The first one is the Haupu Range. This little mountain range could look very humble, very small. You could even think that it's a case of a rejuvenation like Diamond Head, but it's very different. In the case of the Haupu Range, these are one of the oldest lavas from Kauai. And as part of the oldest lava, they're old enough that some species, like the Orson Welles spiders, have developed local endemics to only this mountain range. Another mountain range that appears to be falling right into the ocean is the Napali Coast in the north coast of Kauai. In this case, what happened is the wave erosion coming from the north swells has created these steep erosive patterns on the mountains. If you look at other areas in the north side of the islands, like North Shore on Oahu or Pe'ahi in Maui, these are the areas where normally the big surf competition happens because the big winter swells are hitting. The Waimea Canyon is another example of a very spectacular erosive event. In this case, it is not produced mainly by the river running at the bottom, but produced by another factor. So if you can watch my camera, with my hands, I'm doing what would look like some sort of paleo Kauai volcano. Eventually, this volcano erode, but only half of it. So then, with time, it gets filled up with lava again, and each of these half has about about 1 million year difference on the age. So if you're in one rim of the Waimea Canyon, you're in one age, and in the other one is 1 million year difference. So this difference on the age of these lavas, in some way, made it difficult to make them very concrete or compact. So eventually, they just separate and create this gigantic canyon, a feature absent in a young island like the Big Island. Also related with this high elevation, there is the Alakai Swamp, the highest wetland on the planet. And related with that wetland, I would like to now focus your attention on the rain patterns on these islands. So I already described roughly the geology. Now the Hawaiian Islands are facing the trade winds blowing in this direction, and they create a very steep patterns of rainfall within very small areas. And perhaps one of the most dramatic ones are in Haleakala, where you compare what is the kind of called the Hana Coast with rainfall of like 7,000, 6,000 millimeters per year. But behind that, we can have 700. So one order of magnitude difference in less than 100 kilometers. So again, the same way as the elevation create different habitats, the differences in rainfall also creates different habitats, compacting all this environmental diversity in very small areas. What I will be talking here mostly is about the diversity that lives in rainforest, located between 1,000 to 1,600 meters across the islands. Something that is very characteristic of this rainforest is taxonomically at higher levels, so let's say family and genera in some cases, they're very homogeneous. You will find the same families and genera across the islands in general. But at the species level, sometimes you can find endemicity at the level of a volcano. So you might well know your Hawaiian families, and you can get around at the family level pretty well in all the islands. But at the species, pretty much every place is a different story. So that's where diversity appears. Another interesting thing related with rainforest and age of the island is as islands are subsiding, the amount of area exposed to the inversion layer, so where the clouds sit, is shrinking with time. So in the case of the volcanoes Mauna Kea and Mauna Loa on the Big Island, we see that they stick up of the inversion layer for almost 2,000 meters. So we have 2,000 meters of alpine environment and all the rainforest down here. For Haleakala, it's a little bit less, and we have all the rainforests, especially on the windward side. West Maui does not have alpine environment, and all the way to the top is rainforest. Indeed, Pukukui being one of the wettest places on Earth. And the Ko'olau Range on the back of Honolulu, we see that the inversion layer kind of covers the top of the island, of the mountains. So keep this idea in mind. As the islands get old, they are sinking, and the amount of rainforest area also is shrinking. So this is the final slide of this kind of intro, where I really like it because this cartoon shows you different volcanoes with their ages, but also the distance. So between the Big Island and Oahu, there's like 300 kilometers, but between Oahu and Kauai, 150, but then between Kauai and Kure, about 3,000. So the distance between the islands are also a reflection of a combination of how fast the plate is moving and how active is the hotspot. So that also comes into play. So all this scenery that I just described is where perhaps one of the most amazing life forms have evolved. And in this drawing, you can see illustrated with very good detail, many of the species that make Hawaii one of the jewels, biological jewels of the Pacific, I would say. All right, so I started talking about adaptive radiations, and then I took a detour to talk to this big introduction of how Hawaiian Islands evolve. So how these two concepts are related, and perhaps the relation between them could be illustrated on this observation done by Rosemary Gillespie and Bruce Baldwin, both from UC Berkeley. Rosie, evolutionary biology, focused on invertebrates, and Bruce Baldwin, a botanist. They both found that if you graph the number of species standardized by area against the age of the island, for many of the groups with large number of species, so the ones that experience adaptive radiations, the peak on diversity happened on middle-aged islands in animals and plants. But the groups with smaller number of species, they tend to increase in a more monotonic way, getting more species in other places, which is kind of like a null model, what you would expect. An old place, more species. But why there's a higher peak on diversity on middle-aged places? Remember, this is standardized by area, so we're controlling by that factor. But why there's more species there? So if I sit on the Big Island for 5 million years, would I see the diversity expand and then eventually after 3 million years shrink again? And here is where the chronosequence of Hawaii might be informing about the path of adaptive radiations. So what I'm going to propose now is a very simple conceptual model as to how these two things can combine. So let's imagine we have an island with a patch of rainforest and one species. Because it's a volcanic island, eventually we have a second volcano. Our species colonize this second volcano because there are lava flows. There will be some dynamic of separation and a mixture like the Kipuka on the Big Island. Same process happened for the third volcano, same situation for the fourth volcano. Then we end up with an island with 4 volcanoes and 4 separated populations of a species of something. If the island is big enough, these 4 species eventually might turn into 4 allopatric species. By the lack of gene flow, they just speciate. So then diversity goes from 1 to 4. As the island gets older, some of the erosive process, subduction, might destroy some of this environment, so our diversity will drop from 4 to 2. Now, more interesting things could happen, and organisms could move around within the island. So they, after the extinction of their endemic habitats, they survive in a second place. Now, this sounds, at least to me, sounds like a good idea. What's the correlation with real species? For instance, with the spiders, we found the Orson Welles genera, which closely tracks the geography of the islands on the number of species that they have. There are the Hawaiian stick spiders that they do the same, but they also do something else, which is within the same island, they will evolve different ecologies. So here we talk about non-adaptive radiation, just tracking geography. Here we talk about adaptive radiation, tracking geography, plus evolving different ecologies within a volcano. Another prime example of adaptive radiations are Tetragnatha spiders. The Hawaiian Islands concentrate about 10 or 11% of the diversity on this genera, 360 species worldwide, and Hawaii is 10%. So if you put in perspective the size of Hawaii and the rest of the world is truly a hotspot. They are the result of two independent adaptive radiations. One of them, the web building clade. These spiders spin orb webs like any other Tetragnatha in the rest of the world. This group has the highest number of species and has been studied the least. Then we have the spiny leg clade. They don't spin webs, so they have this ecological shift. They are less species, and they are better studied. This group, on top of that, has evolved 4 different ecomorphologies. So ecomorphologies are an ecologic syndrome where a species has some set of traits that match their ecology. In particular, we have the green Tetragnatha, which lives under the leaves, the maroon that lives in moss, the big brown that lives in bark, and the little brown that lives on twigs. We have these 4 ecomorphologies evolving semi-independently in all the islands in a repeated fashion. We never find 2 species of the same ecomorphology living together. The idea is 2 species with the same ecology might enter in competition, and that will prevent their coexistence. This pattern of 1 ecomorphology per site is repeated in other well-studied adaptive radiations like the Caribbean anoles, the East African cichlids, etc. But on the volcano of Haleakala in Maui, this rule seemed to bend a little bit. So in Haleakala, we find 3 species of the green ecomorphology, but on different places of this big volcano. If we look at closely, here we have the 3 localities where we find Brevignatha, Waikemoi, and Macracantha. They also have a second population on another volcano or another island. These 3 species are closely related and have the same ecology. So here is the question, how did it happen? How did they reach this distribution? And what we can learn about the general process of adaptive radiation by answering that question. So the first hypothesis to explain the current distribution would be the following. Let's imagine before the origin of the big island, we have an ancestral species with 3 populations, Lanai, West Maui, and Haleakala. By allopatry, they turn into 3 species. Then they move around and reach their current distributions. As you can see, this is a more applied example of my cartoon model at the beginning. Another hypothesis is they all evolved in the same volcano in Haleakala with parapatric speciation. So ancestral species with 3 isolated populations turn into 3 species, then they move around and reach the current distribution. These 2 hypotheses are extreme cases. Most likely, you can imagine intermediate cases where parapatry happened for 2 species and a third one evolved in allopatry somewhere else. Also, you can imagine more complex scenarios where all the species colonize all the islands. So if we look at the genetic makeup of a species, let's say on Lanai, we might find some contribution, some genetic contribution of the other 2 species, but it's just 1 species, the one that established prominently. There are also other strange details like the fact that there's no green spider in Molokai and never been found. So how do we test this hypothesis? The best way to do it is using population genetic methods, because population genetics allow us to see what are the source populations, what are the affinities between species, etc. So I'm not going to spend much time on the technical details, but I'm going to give you a big overview. And if there are more questions, I'm very happy to answer them. So what I did is I apply a method called Exon Capture. Here you first, of course, go to the field, collect in different localities, do species identification, DNA extraction, and you prepare a genomic library. In parallel, you sequence the transcriptome of 1 specimen of those species, and from that transcriptome, you select pieces of DNA corresponding to genes. And those genes are the ones that you end up selecting as probes. What do I mean by probes? Imagine that you have your genomic libraries for your 100 specimens, and you have a selection of probes of 10 genes. So you want to see what is the version of that each of those 10 genes on each of your individuals. So that's how you kind of pull or capture that fraction of the genome for each individual that you are interested in. We did that using something called hybridization. after hybridize, so capture those genes that we're interested for each individual. We run some set of post-capture, post-hybridization controls to make sure that the experiment worked. We send that to sequence in a high throughput machine, Illumina, which allows us to sequence a huge amount of DNA. Then we run several filters to make sure that we have good quality data. After that, using a program called ENGS and side frequency spectrum method, we can finally use or generate the type of analysis that classically are done in population genetics, such as admixture mapping, principal component analysis, or phylogenetic networks. So a little bit of detail, technical detail, and then we move on. So our probe or array design had 1.7 megabases, equivalent to 1.8 ORFs or open reading frames. Here, there's an illustration of our samples. We barcode them with pieces of adapter DNA that then we can demultiplex or separate bioinformatically. So we pull them all together, we run the experiment, and then after the sequencing, bioinformatically, so in the computer, we separate samples corresponding to each specimen. Just to give you an idea, okay, we do that. Then we run the controls. There's the positive control where we want to see that there is enrichment on the genes that we are interested. There's the negative control where we want to see that there is depletion on the genes that we are not interested, because if there are too many genes that we are not interested, we're going to lose sequencing power. We also check the size distribution of these pieces of DNA to make sure that there's no PCR duplicates or any other kind of strange DNA sequence that will, again, reduce our sequencing power. We did 3 experiments, each one yielding about 30 gigabytes of data. This is in the format of FASTQ, which is similar to a text file. So you can imagine 30 or 90 gigabytes of data of text file is a huge amount of data. That's great because it allows very good resolution. Here, just to show you that it exists and I can talk about it, here's the pipeline of the different programs that we use to process the data, assemble the capture, assemble the capture segments, and then all the quality checks running up to the final data analysis. Okay, so that was the technical part. Let's go back to the biology. So remember, these are the species. Our question is, how do we reach this distribution? So the first analysis is a principal component analysis. Principal component analysis basically groups things by genetic similarity. And the way of how you read this is as it follows. Each point is an individual. The shape of the point represents the identity based on locality. Remember, we only find one species per locality so far. And the color is the identity based on morphological identification. In gray, we have juveniles, which we cannot distinguish between these 3 species. So if something is juvenile, we don't know what's the species. We have 3 species, so we hope to find 3 clusters. And here is where the first surprise is. In green, we have 2 clusters. And if we look closely, each cluster kind of makes sense. So we have green circles, so they are Brevignatha morphologically and by locality, and we have gray circles, so they are Brevignatha by locality and they're juvenile, and genetically they all cluster together. So these 2 clusters happen to be the specimens from the Big Island and from Maui. So what was once called a single species, Brevignatha, with 2 populations, really genetically correspond to 2 species, 2 cryptic species. So here's the first result. Then we move on to the next group. Here's orange Macracantha. By locality should be a square, by morphology should be orange. So we have an orange square, perfect. We have a gray square, so that's a juvenile of Macracantha, very good. And we have this diamond, which is a new record. So we did collections in Kolau Forest Reserve in Maui. We didn't know what to find there. There's no previous record, so we didn't have a prior. But by looking at the morphology, like in this case, we realize it's Macracantha. So basically here we have a compact group of Macracantha specimens. Remember, those come from Lanai and Maui. Things get really interesting when we look at Tetragnatha Waikamoi. This is blue triangles. So we have a blue triangle, good. We have a gray triangle, good so far. But then we have a blue square. That means that morphologically is Waikamoi, but it's in a locality where you hope to find Macracantha. We also find gray circles, which means it's a juvenile. Morphologically, we cannot tell what it is, but it's in a locality where you hope to find Brevignatha. If we do a PCA analysis just with these specimens, we can see the separation between West Maui and East Maui, and that it is in East Maui, in Haleakala, where we find these Waikamoi individuals showing up in areas where you hope to find the other species. So here, the second big result, Tetragnatha Waikamoi is actually coexisting with the other species. So the next question would be, is it competing ecologically, and is it hybridizing? The competition question we cannot answer because we are already in the lab, we don't have the specimens, but the hybridization could be answered. And for this, we use this admixture plot where we show all the different populations, and basically each bar is an individual. If you have a bar with two colors, it means that it has a mixed heritage from two different populations. The interesting ones are Kipahulu Valley and Lower Waikamoi area, which are the areas where we found two species coexisting. And here we see in both cases two colors, but two colors but two colors with solid bars. So there's no hybridization on these individuals. So third result, they are coexistent, they are not hybridizing. So they are already well-established species. We get the same result by actually calling SNPs instead of using site frequency spectrum, which is a way of treating the data. We treat the data differently. It's the same data, but using different tools, and we generate this phylogenetic network. Here we find the divide between the Big Island specimens and the ones from Maui. This is congruent with what we've seen before. We see Brevignatha with the two very well-defined cryptic species now. Interestingly, Macracantha, which in the principal component analysis appears as a single cluster, now splits in two groups, Lanai and East Maui or Haleakala, which biologically makes sense. And Waikamoi, which has populations on West and East Maui, also separates, but has a common root. So biologically makes sense. What matters here is that the individuals of Waikamoi that showed up overlapping with the other species cluster with the rest of the species, and they're
Hello, everybody. It looks like we're having some technical difficulties, so why don't we just wait a minute and see if Darko comes back on. It looks like he is reconnecting now. Hi, Darko. Oh, you are muted. You should be able to unmute yourself.
Can you hear me now?
Yes.
Great. So I think I disappeared when I was talking about Waikamoi. Is that correct?
That sounds correct to me. So.
Okay, so I'm gonna start a little bit before there, just in case. So apologies for this problem with the connection. What I was saying is, with this phylogenetic network, we look at in Macracantha two different populations, one's in Lanai and the other on East Maui. Did you hear that already, Hailey?
I think that's a good place to start.
Great. Okay. So we find this this biologically congruent result that we didn't see with the previous type of analysis. And also we find that in Waikamoi, there are two populations, one on West Maui, the other on East Maui. But the most interesting thing is the one from East Maui really has all the specimens together. So the ones that are overlapping with the other species do not show up in the middle as hybrids. So this is congruent with the idea of no hybridization. So what we learned? So from all of this, we learned that the three original nominal species cluster in 4 or maybe 5 clearly divergent genetic lineages. Also, the populations from different islands of Tetragnatha brevignatha seem to be genetically as divergent as what happened between different species. So we have 2 species instead of 1. Tetragnatha waikamoe is a widespread species present in different parts of East Maui or Haleakala, and more importantly, there's no hybridization between closely related species. So what this all means, if we have to summarize, this data is telling us that we can detect sympatric distribution of closely related species, so species overlapping that are similar, that are genetically close, from the same ecomorph, so with the same ecology, without hybridization. Why this is so why this is so relevant for adaptive radiations? Because before this and other data, it was thought that different ecomorphs should be separated and never could occur. So thanks to the chronosequence display of the Hawaiian Islands, we were able to find the exact snapshot where species have already formed, but they come into secondary context. But because they're already well-established species, even if they have the same ecology, they don't hybridize. Now, the discovery of same ecomorphs coexisting is something that in recent times with other well-studied adaptive radiations like the Caribbean anoles or the East African cichlids appear to be more common than we thought at the beginning. So our finding with the Hawaiian Tetragnatha comes fit this new set of data to understand the process of adaptive radiations. So how the model would look now. Remember, we have the island with the volcano and the species. Then the island grows. We have 4 populations that turn into 4 species. And then these 4 species, eventually, some of them move around, some of the localities disappear, and then they reach this secondary contact. This is exactly the moment that I'm showing you with the spider data. Now, from here, what could happen next is that these 2 species with the same ecology might start interacting, perhaps competing, and that will create character displacement at an evolutionary scale. So character displacement is a mechanism observed in wild species where 2 close related species come into competition, they morphologically differentiate, so they reduce the competition between them. So the same thing might happen between the spiders, and that will create different ecomorphs. So instead of both living under the leaves, one might start living on the bark or moss, etc. So that is how we think from a common ancestor, due to geography, new species will arise, and then after they come into secondary contact, this character displacement at an evolutionary scale might give rise to different ecomorphologies. Now, I want to repeat that what we show with our data is the formation of new species and coming into secondary contact. So this point. This next point, the demonstration of this is something that is yet to come. So with all of that, I just want to finish saying mahalo. Thank you very much to so many people who supported this work, starting with my PhD advisors, David Limber and Rosemary Gillespie, many people in the field helping me, giving me rides to different places, teaching me about Hawaiian biodiversity, many of the permitting agencies that are protecting the land and then allow research. Special thanks to Hawaii Experimental Tropical Forest. A lot of my data for Mauna Kea was taken in Laupahoehoe. Also had the chance to collect in Pu'uvava, but didn't find, it was not the right habitat for this type of. So thank you so much for your help to all these agencies. Also, thank you to people who helped me with lab work and data analysis at UC Berkeley and several sources of funding. Having said that, if you are interested on this, there is a publication already which describe in great detail or the more technical part.
Hello. It looks like we lost Darko again, just briefly.
Am I back?
Yes, you're back.
Okay, so hopefully it's not going to happen again. What I was saying is, if you're interested, there is a paper already published with this and a blog about this. And currently we are finishing a focus on population. So hopefully in the next month, there will be more results exploring in great detail the population structure of the spiders within the big island, within this cradle of evolution. So having said that, my apologies for all the connection problems, and thank you very much for your attention.
Yeah, this was a really, really fascinating talk. I really loved how you went through and explained kind of the backstory and then kind of went into the work that you're doing. With that, I don't know if I saw any questions in the chat. Just lots of thank yous and mahalos.
I have a question, Haley.
Yeah, go ahead.
Yeah, so Darko, you mentioned that you have your publication out. Could you put the link to that publication in the chat, please?
Sure, sure. So
yeah, I will also, I always send out a follow-up email after the talk, and I will include that link in my follow-up email. Just
that will
if
work.
anybody...
That will work fine, and that way Darko doesn't have to do it right now.
Okay.
Okay. So let's do it in that way.
And then what else? Will it be possible to watch the recording? Yes, I will be uploading it to YouTube at the end of the week, and it will be in a YouTube playlist that is already set up, and I will share both the link to the publication and the link to the active YouTube playlist with everybody at the end of the presentation. But other than that, I think you're just getting a lot of excellent and fascinating talk. This is really interesting. Lots of thank yous. Looking forward to diving into your publication.
Great.
So yeah, if anybody has any questions, now is your time. Yeah, and thank you to everybody who is privately messaging me and thanking me for hosting this series. It really means a lot. You're all very, very kind. We'll wait just a few minutes and see if there's anybody with any last questions, just to give them the chance to speak. But other than that, it's been an honor to host this for you guys. It's been an honor to get to talk with so many amazing scientists doing really fascinating work. And I am very glad that I was able to reach so many people and to meet so many people. Yeah. So waited a few minutes. I think with that, we're going to end. Thank you all so much.